Hello, this is Kang from Professor Hyungsuk's laboratory in School of Mechanical Engineering, Yonsei University. I would like to talk about the high-resolution acoustophoretic 3D cell patterning for disease treatment. Related work was published in Nature Communications, December 2018. Further information of this presentation can be found in the article and video abstract, of which the links are in the description of this video. Protein fibers and cells are arranged into tissue-specific geometric patterns. Since their biochemical and biophysical interaction plays a critical role in biological functions, including stem cell differentiation, it is required to mimic the microgeometry of the tissues. Bioprinting techniques are able to print cell-laden hydrogel solution. Recently, researchers have tried to fabricate artificial tissues to mimic various types of tissues and organs in our body using the bioprinting technique. However, bioprinting technique has some technical issues. Spatial resolution for the bioprinting depends on the nodule size. However, reducing the nodule size can cause a higher shear stress on cells and cause cellular damage or cellular death. Also, cells are just mixed with the hydrogel solution without any external forces to aggregate them. So, it is difficult to induce the cell-cell interaction immediately after the printing. Because of this characteristic, when using bioprinting, it is not easy to simulate a tissue in which cells are arranged in a specific geometry at the scale of some millimeter level and cells are densely clustered. In our body, circulation of nutrient, oxygen, and waste is governed by our blood vessels, and the blood capillaries plays a critical role in the mass exchange between the tissues and blood vessels. They are highly aligned in our body and consist of cells which are aggregated. They are spaced with a distance less than 100 micrometers. Thus, using the conventional bioprinting technique, it is not easy to simulate the structure of capillaries. Here, we apply the acoustic field to translate the cells to the potential well, and they are placed in proximity. We developed the acoustophoretic device in which a disposable cartridge can be mounted on and detached from the device. Cells were patterned within 10 seconds inside the hydrogel solution, and the hydrogel solution was then solidified. After the solidification, the cartridge was detached from the device, and the patterned cells in the cartridge was then cultured in the incubator and transplanted into the mouse model. Right after the gel solidification, the patterned cells showed similar geometry to the blood vessels in mouse muscle. From the cellular experiments, we found that the cell patterning using acoustic wave could increase the various cellular functions related to the generation of new vessels and stability of the blood vessels. After the fabricated tissue was transplanted to the mouse model and cultured for 1 week, we injected the fluorescent dye solution FIT-dextran into the blood vessels of mouse tail. The fluorescent signal was only shown in the aligned group, not in the random group. This indicates that the patterned cells form perfusable blood vessels and connected with the pre-existing blood vessels in the mouse. In addition, we evaluated the recovery of the blood perfusion in the mouse leg by the transplantation tissue, which was indicated by red color in these Doppler images. We found that the recovery of blood perfusion was highest in the aligned group, and the loss and the necrosis of the mouse were reduced in the aligned group. We expect that our acoustophoretic tissue fabrication technique can be applied to fabricate nerve bundle, skeletal muscle for disease treatment, or to make the lab-grown meat. Thank you. Further information about this presentation can be found in the article and video abstract of which the links are in the description of this video.